Today we'll talk about the fundamental tissue and mitosis. We will have three parts of this. The first part has to do with a virtual microscope, how to identify things from the images by using the computer. Describe the steps in tissue processing and discuss the different types of microscopy, both light and electron microscopy. Then we will interpret cytologic features of cells by different staining, differential staining, with hematoxylin and eosinophil remain stained, but we also use PAS and toluidine blue, as well as processing tissue for conventional transmission electron microscopy as well as scanning electron microscopy. Part 2 has to do with recognizing and identifying cells using the microscopic preparations that we have, the images that we have, a little bit about how to observe the different structures in the images that we have, and to understand the relationship between structure and function of cells. Part 3 has to do with mitosis, talk about the stages of mitosis and cellular structures that are associated with mitosis. Mitosis is a process of cell division by which you produce two daughter cells identical to the previous parent cell. Enjoy. Fundamental tissues and mitosis. This is part 3. We'll be looking at images of mitosis. So we'll be identifying the mitosis and cellular structures associated with mitosis in part 3. Mitosis is cellular multiplication, where cells divide and have two identical cells produced just like the original cell. So this is what happens in cells to duplicate the numbers of identical cells. This is what happens in the beginning of spermatogenesis and the beginning of oogenesis, and then somatic cells of the body have mitosis. This is the way organisms grow. Cancer is essentially uncontrolled mitosis. So cancer is not a new thing, it's the same thing, it's just out of control growth. We will look at the onion tip of an onion ring and also some fish eggs to identify the stages of mitosis. If we look at the onion tip, and you can see these little boxes, these are cell walls for which the word cell was coined. And here we can see interphase nucleus. Interphase nucleus got a lot of euchromatin and a heterochromatin in a nucleolus. So that's your typical interphase nucleus. Now, whenever you get prophase, the chromosomes have condensed, the nuclear envelope is broken down, and so the chromatin material is forming these chromosomes. So the DNA is lining up in the chromosomes we see here and here. And then whenever they're lined up, that's in metaphase. So what was getting together is lined up in here, and here we can see metaphase. When they start to pull apart, as you see there and there, that's anaphase. There, usually anaphase, you can see that the chromosome, the back part of the chromosome is lagging behind. It makes little V's. You know you're in the anaphase then. And then finally, whenever they are pulled apart and maybe cell division has occurred, that is the complete cells are two different daughter cells now, then you're in telophase. So these are telophase that we see. We can see the same thing in the fish eggs. This is the interphase nucleus here and here. And then this is the prophase, where the chromosomes are forming from the interphase nucleus. And then this is metaphase, and you can see where they have lined up very nicely, as lined up there. And these structures here and here is the spindle fibers, which are the microtubules that are associated with pulling apart, and this would be where the centrioles are located here and there, which organize these microtubules that can pull the chromosomes apart. Then anaphase, again you get little V's there, ends of the chromosomes lagging behind, and you get the centrosome area where the centriole will be located here and there. And then when it pulled apart and you get complete cytokinesis, here the cytoplasm is separated, now you're in telophase. And here we see the nucleus. This is a interphase nucleus here with euchromatin, heterochromatin that we see of the nucleolus. The cytoplasm here, this is the nucleus. And you see there's no nuclear envelope during the metaphase. This is going to be metaphase. We're going to pull it apart. And these are the centrioles that are connecting to these microtubules that are attached to chromosomes that are going to pull them apart. So this is metaphase all lined up. And that brings us to centrioles. What's the function of the centriole is that they are the organizing center. They organize the microtubules that pull the chromosomes apart. And centrioles is composed of 9 triplets, microtubules, and no central pair. So these are microtubules, stable microtubules, attached to proteins, and they're microtubules, labile microtubules, pull the chromosomes apart. Also, centrioles are associated with cilia development. It's the source of the basal bodies that produce cilia and also flagellum of sperm. So you might think that since cancer is uncontrolled mitosis, you might be able to use antimitotic drugs. But unfortunately you can't do that because you have cells like in your GI tract that undergo mitosis routinely, and they're replaced every 4 to 6 days. And so that's why someone getting chemotherapy gets sick, because the chemotherapy is actually killing some of the cells lining the gut as well. So it's not easy cure for cancer, and certainly you can't use Taxol and some of those drugs to just kill all mitosis in the body, because some mitosis is required. Use your atlas is one of the things that we want you to do to be able to identify things. Here we see some pancreas and blood and stomach and testes. They all look different, but you can do this. This is not the atlas that they recommend for the med school. Use the book they recommend. The end. I hope you enjoy and I hope it's useful to you. Thank you.